Yes, hello everybody. So many thanks for the invitation to this conference. It's my pleasure to show you our work on the genetics of COVID-19. And before I start, I just wanted to briefly show you our institute here in Kiel, which is here in the north of Germany, close to the border of Denmark. So this is our nice research building. And we are operating really a large genomics facility here. We're one of the 4 main sequencing centers here in Germany. You may not have heard of Kiel. That's not surprising. It's a small city and it really feels a bit like these guys here, but sometimes also small guys can do some, great things, and I hope I can show you some of our exciting research we did on COVID-19 in the past. I don't have to introduce you, of course, to the disease. The pandemic is affecting all of us, the entire world. These are the most recent numbers at the time of this presentation. So we have over 4.7 million deaths due to COVID-19. And what I wanted to point out here is also the really large death number here in Italy and Spain, especially during the first wave in 2019. I will come back to this. Now, what are the risk factors for COVID-19, especially severe disease course of COVID-19? Well, as you know, age is the most important risk factor. So the older you are, the more likely you will get COVID-19 and also a severe disease course. But also gender is a very important risk factor here. Males are more often affected by COVID-19 than females. Now, what about genetics? Well, at the time that we started our study here, and also here as part of this epidemiological study, genetics was not investigated as a risk factor. That's why we wanted to run a genetic study. Now, why so? Of course, because we want to learn more about actual disease causes. We want to explain why some people are more susceptible to the virus and why some maybe develop a severe disease course. And of course, to help in the identification of evidence-based candidate targets for downstream drug research. Because many of the treatments that are used to treat these really severely ill patients, these are really off-label treatments. Originally, these treatments were developed for example, malaria or Ebola. And now they also seem to work in treating severe COVID-19. But probably one can do better by using a more evidence-based candidate. And there is really good support from the literature that if genetic evidence exists, that this really supports the drug discovery and development. Because these drugs are twice as likely to be successful in clinical development. And this has been shown before and published by Matt Nelson and colleagues. And indeed, if you look at the most prescribed drugs in the US, for 9 of 17 targets here, there is genetic evidence, which just shows you that these kind of studies are really helpful. Now, genome-wide association studies, or in brief GWAS, I think I don't have to summarize this method because it's very popular these days and it was very successful, of course. It is still the state-of-the-art method to identify actual disease causes in especially complex traits. And you normally do this by collecting a large number of unrelated control samples, unrelated case samples. Then you perform SNP array genotyping, and then you do just simple statistical association testing. So do you find certain alleles or genotypes that are statistically more frequent in cases as compared to controls, or the other way around, more frequent in controls than in cases, if you're looking for protective factors. And of course, there are many advantages of the GWAS approach. They are relatively cheap and high throughput, even cheaper, of course, and more high throughput than whole genome sequencing, for example. And for common variants, this approach works really well. But there are also a lot of disadvantages. You really miss out on the rare variants and non-genetic factors and so on. Now, GWAS have been very successful. This just shows you that for a lot of traits, we have discovered genetic factors here. This summarizes here 10 years of GWAS research. So the human genetic architecture is becoming more and more complete here for many diseases and traits. And once you have a disease gene identified or a locus identified, you really have a good hypothesis to test in experimental research. And this is just a summary from our work in inflammatory bowel diseases. That's actually the main disease we're working on here in Kiel. And just shows you that before the first disease gene was identified in 2001, no one really worked on NOD2, which is the main disease gene in IBD, or on autophagy. And when these genes were identified by GWAS, there's a lot of targeted research and discussion in the scientific literature. So it really gives you a very good starting point for the next studies, and that's what GWAS are good for. Now I'm coming to our GWAS study for COVID-19, and just to remind you that the outbreak in Italy in last year was really severe, and so the first patient really here arrived in February in Rome, and then until the end of March here, the disease had spread to the entire country of Italy. So this country was really overrun by the virus. And many of you remember well these really nasty photos here. This is from Bergamo, a city in Italy, where they had to transport out the bodies, the coffins, because the city was overwhelmed with the large death number. And so they had to bring them to other cities. And it looks a bit like a war scene here. So we don't want to see such things, of course, again. And that's why everyone, of course, should get a vaccination shot. And at that point in time in Germany and also Norway, we didn't really have many cases yet. And so we had the luxury position here to think about research. What can we do really to find out more about COVID-19? And Tom Hemming Carlsen here from Oslo, a hepatologist, he really convinced me that we should do a GWAS study on COVID-19 and in particular severe COVID-19. And we received generous funding from this benefactor here, Stein Erik Hagen, one of the richest men in Norway, to do a first GWAS. And I should say that Tom also had really good contacts to Italy and Spain, to clinics which were really flooded with patients in the intensive care units. And with support of the clinicians there, we collaborated with 3 sites in Italy and 4 sites in Spain. We were able to collect DNA samples of over 1,500 patients with severe COVID-19. And this figure here from our article summarizes the whole study and just shows you that at the peak of the first wave, we started the study and we were able to complete the study within just 3 months. Okay, so it just shows you the power of GWAS, how fast you can be from start to the end in identifying genetic risk factors. This is what it looked like in practice. You see here, of course, the samples arriving in our lab. And here's a big shout out to the clinical and the clinical team in Italy and Spain, because even though they had really these war-like situations, very bad situations in the hospitals, they still recruited patients into the study, collected the blood, and sent it to Kiel with some nice messages. I mean, you can see here one of the packages to Kiel. Everything is going to be all right. And so a big thank you to the people here. I think you did something very important here during a difficult time. Now, this is a summary of the study cohort. You can see on average we had elderly people above 60 years, and we had predominantly also males among the severely affected patients. And I should say that we defined severe COVID-19 by that people had to be in the hospital, they had to be given at least oxygen ventilation. Many of our patients also were connected to a ventilator here. These were the more severe affected. Well, when we performed the first association analysis, we were actually surprised what we did not find. I expected, for example, an association signal on chromosome 6 in the HLA region or the MHC complex, because this is such an important locus for our immune system, and it's associated in so many autoimmune and infectious diseases. But there was no signal at all, and until today, I have not seen any convincing HLA study yet for COVID-19, so it seems not to play any role when it comes to increasing susceptibility for an infection or a severe disease course. This is the Manhattan plot of our study here. The more peaks you have, the merrier. We here had 2 prominent peaks, which passed the magic level of genome-wide significance. One on chromosome 3 and one on chromosome 9. And the signal on chromosome 3 was very, very strong. Now I'm going to summarize now briefly these 2 loci. Here you see the association results. As I said, genome-wide significance in our study cohort. And if you look at the odds ratio, we had a really strong odds ratio here, especially if you correct for age and sex, over 2 for the chromosome 3 variant and about 1.4 for the chromosome 9 variant. And especially this variant here, it's a very strong association. As you know, normally in GWAS you have values around 1.1, 1.2. So this is a very strong susceptibility locus. And if we look at the genotype distribution, we also observed that patients who were homozygous for the chromosome 3 variant were significantly younger and had a more severe disease course than the others. So this seems to be really a severity locus. Now we published this very early in a preprint journal, and the peer review was also conducted very fast, and the paper then appeared online in the New England Journal of Medicine last year. So it was quite a big success for us. Over 800,000 page views happened since then, and we were really proud that the New York Times and also over 500 other newspapers highlighted our first genetic study here on COVID-19. But everyone was focusing on the chromosome 9 locus, the blood group, association, although the chromosome 3 locus, as I said, is a lot stronger. Anyhow, I will discuss these loci in a minute. And yeah, there are also some highlight papers, and I should say that in retrospect, the German government also gave us some funding to conduct this study, and they also highlighted our study here in a press conference. So we were really proud of this and happy with the outcome. Okay, chromosome 3. Well, this is what the signal looks like, a typical regional association plot. And you can see that underneath the peak, there are really several candidate genes. And you cannot unambiguously assign a single gene here. In fact, the credible set of variants that are most relevant here is a bit larger. And we had at least 4 very interesting candidate genes. And given the strong odds ratio, I like to call this region the not 2 of COVID-19. So what not 2 means for Crohn's disease, probably this locus means for COVID-19. So it is a strong association signal. Unfortunately, it's not really clear yet. Is it a combination of variant? Is it a combination of genes? Or is it a single variant, a single gene? Nevertheless, people are performing focused research now on the different candidate genes. So I'm really looking forward how this signal gets further dissected. And it got replicated by others. The odds ratios are a bit smaller. That is normal because, as you know, the discovery study has this winner's curse. So the odds ratios tend to get smaller over time because it's just logical that in the first study it's so high because it is high, otherwise you wouldn't have detected it. So, but it seems to be really a locus for disease severity, and this has been replicated by many other groups. Now, the second signal on chromosome 9 is quite exciting because this is directly located in the region that encodes for the ABO gene. And ABO, you know, is one of the most important blood groups. Again, there were many SNPs here in the credible set after fine mapping. And what we also saw is that if you translate the SNP genotype data into the blood group data, you also see an association on the blood group level. So that we assumed maybe blood group really is associated here with the disease. And I have to honestly say that we were not the first to report an association between ABO and COVID-19. There were actually, back in time, there were 2 preprints, and one of the preprints was also accepted after our study was published, that found this association on the serological level in a smaller set of patients, and but still they see the same trend that blood group O is associated with protection for infection, and blood group A is associated with a higher risk for acquiring COVID-19. But these are serological studies, as I said, and we found really this convincing and statistically robust association on the genetic level. Yeah, we see the same trend. Blood group A means risk and blood group O means protection. I think it's difficult to make a conclusion for AB because it's such a rare blood group and we simply lack statistical power here, but B does not seem to be associated. Now, why blood group ABO? Well, we don't know. Yeah, we don't know, first of all, is it a SNP association? A SNP could impact a functional element and, of course, have a regulatory effect on something in this region. There could be also, of course, an association of the blood group itself. And here there are different explanations. Some said, well, It's known that if you have blood group A, coagulation is increased, and the increased coagulation is also a high risk factor in COVID-19 for a severe disease. And I also like the other hypothesis, which could explain why blood group O is protective. If you have blood group O, you lack the A and B antigens on your cell surfaces, and you have antibodies against the A and B antigen in your blood. And as we know, when the virus is replicating in the host, in the human host, it uses also host material in the shedding process. So sometimes you find host proteins on the surface of the virus. So if the virus comes from an A or B host, it may have A or B antigen on the surface, and then it can be detected because you have anti-A, anti-B antibodies. So that's one of the explanations that is made why blood group O may be protective for some viruses. But it's not proven yet. Well, I should say that when we published the preprint article, we received quite some heavy comments through the social media, especially on the blood group association. People, for example, wrote on Twitter, hey, no kidding, you'll find an association with ABO as you use blood donors as control. And blood donors are preferred by transfusion medicine departments because they have blood group O, and these are all universal donors. Okay, and yes, we used to some degree blood donors, but we actually could prove, and this is just one of the proofs we provide here in the supplement of the articles, that if you're recall donors, so someone who comes in regularly and where you may have a bias towards people with blood group O, and if you compare this to first-time donors, so where there should be no bias towards a specific blood group, we do not see any frequency difference at all. Some people also said, hey, there is a age bias here. It's known that the blood group distribution is different in different age groups. This is, of course, nonsense, and we could also prove this here just by looking at the data from over 100,000 blood donors at our university hospital. So it was actually good to publish our article so early in a preprint journal, because you get a lot of additional input through the social media and so on. I was a bit skeptic here before, but in the end, I think we did the right thing. So it was quite helpful, and we could address all the comments then for the final manuscript. What makes me also proud and happy is that our findings were replicated by others. This is the Manhattan plot from the 23andMe trans-ethnic GWAS, and you can see again here signal on chromosome 3, but also very strong and consistent signal on chromosome 9 for blood group ABO. And 23andMe also wrote that they found evidence that blood group plays indeed a role. So we really think that our finding is true. And also the Danes, who have a really large cohort here, if you look at the numbers, they see the same association as we see: protection for blood group O, risk for A, and nothing really for the other blood groups. So I consider this association as validated and robust. And ultimately, we also contributed our data to a terrific international effort, the COVID-19 Host Genetics Initiative. And this consortium lately published for a data set of almost 50,000 patients and up to 2 million controls. So it's a major genetic meta-analysis, and here they had 15 genome-wide significant loci, and as you can see, chromosome 3 very strongly associated and confirmed, the strongest hit, and chromosome 9 is also robustly associated in this international meta-analysis. So ABO, I think, no more doubt that this is a true positive association signal. What's not clear until today is whether the blood group association has something to do also with disease severity and not only with increased susceptibility to infection. And this was actually, there's one large study from Canada where they actually showed that depending on what blood group you have, if you're O or B or if you're A or AB, there are indeed statistical significant differences whether you receive mechanical ventilation or how long you actually stay in the hospital. Now, but most studies actually say that if you talk about ABO, it has rather something to do with susceptibility to the infection, okay, and not with severity, while for chromosome 3 it's really a severity locus. Now ABO is associated with so many different diseases and traits. I just wanted to show you here a study from us on hemorrhoids. It's a completely different disease, under-investigated, very common, and we actually completed here the first genome-wide association study on hemorrhoids disease in almost 1 million people, and we described the first 102 susceptibility loci. And guess what? The second most significant association with hemorrhoids was ABO. And placement 3 were other blood group related candidate genes. Here it's the other way around. We have blood group O. This is actually risk for hemorrhoids or hemorrhoids disease. If you have one of the other blood groups, it means decreased risk. Don't ask me why. I can only say what we see here in the association signal. And lastly, ABO is really haunting us. We conducted a large microbiome GWAS, a microbiome quantitative trait locus analysis, and where we correlated stool microbiome data of 9,000 Germans with genetic data of the exact same individuals, meaning we are looking for host genetic factors that may shape your intestinal microbiota. The most consistent and strongest signal here was ABO. So depending what blood group you have, and also if you're secretor or non-secretor, that's another blood group system here, this really was associated with specific microbes that were more prevalent depending on what sugars you in the end have. Because the blood groups, they are not only expressed on red blood cells, but they are also expressed, in the case of ABO, for example, on intestinal epithelial cells. So they are on the surface, and these sugars, they are there also to feed your microbes if diet is not present. And no kidding, there is an association between ABO and specific microbes. And this study here was published beginning of this year in Nature Genetics. You may read further there. We don't have the time to go into details. I just wanted to show you that ABO is a very pleiotropic gene. Now, coming to the end here, in the last minutes, I just wanted to show what's next. Now, since we did find the ABO association with COVID-19, we asked ourselves, of course, are any other human blood groups maybe relevant for COVID-19? And for this, we developed a custom array to genotype blood group SNPs and to derive the other blood group phenotypes here. You should know that at this point in time, there are 43 blood groups known. So ABO is just 1 out of 43. And it's not really clear why we have so many blood groups and what they are for. It's also unknown for many of them, but as I showed you, many of them are sitting on the cell surface, and so they could be relevant also in host virus interaction in infectious processes. And that's why we want to do a systematic analysis here. So far, we didn't find any other association apart from ABO. But what we're currently doing is also resolving the rhesus factor here, rhesus positivity. And also, if you look at ABO, there's A1, A2, and so on. So in this study, we can increase the resolution, and this is something we're trying to write up at the moment. Then we increased the sample size. So besides the Italian and Spanish cohort, we also included a smaller Norwegian and a smaller German cohort. The Italian and Spanish dataset was also enlarged. And here, if we look at the GWAS analysis here, we find 2 additional genome-wide significant signals. And I'm just showing you one of the signal here. This is now out also in medRxiv. It's a large structural variant here on chromosome 17. It's a very large and well-known inversion polymorphism that we find as associated with severe COVID-19. So I really recommend you this article here. It's a very, very interesting polymorphism here. There are also some very attractive candidate genes here below. Unfortunately, we lack the time now to go into more detail here. We're also doing resequencing of part of our cohorts. So we were not allowed to do whole genome or whole exome sequencing. That's why we developed a candidate gene panel. These are the candidate genes we are resequencing. And we are in the process of analyzing over 9,000 samples here from our preliminary GWAS. So only Italians and Spanish patients and controls. And maybe we find also some interesting rare variant associations here in this core. So this is work done in cooperation with Kerstin Ludwig in Bonn, who's primarily driving this. Yeah, finally, I of course want to thank all the collaborators. You see here a very long list of people. So every study nurse, every technician who worked really day and night on the study. Was an exciting time, but I think in the end also a successful time. I want to thank. I especially thank, of course, the study sites in Italy and Spain. These are the people on the top here who locally worked very hard that we could finish the study here within just a few months. And here are some more photos from Italy and Spain from our collaborators. So you can see it was a really difficult time, and even though this all happened there, they took the blood samples and shipped it to the small unknown city of Kiel. So they entrusted us with this for the analysis, and in the end, I think turned out quite successful. Of course, we hope that these association signals are further worked up. You need to do a lot more downstream experiments now to understand really what's going on here in these regions. Yeah, and with this, I thank you for your attention. I also thank my working group here. This is a photo from the good times where you could still do group retreats. Yeah, we're almost there yet again. So many thanks for your attention.